Imagine waking up one morning, looking into your aquarium, and seeing a fish glowing, like really glowing vibrantly in the dark. Not because you painted it, nope, but because its very DNA was intricately woven with instructions taken from a bioluminescent jellyfish.
It sounds like science fiction, doesn't it?
It really does. Or think back to 1996. Remember Dolly the sheep? Big news everywhere.
Oh, absolutely. The first cloned mammal?
Right, but born not from two parents in the usual way, but from the combined genetic material of three different sheep.
Yeah.
And this stuff, it isn't just a quirky thought experiment or, a scene from some future sci-fi movie anymore. This is the astounding, kind of mind-bending reality of genetic engineering.
It really is. A field that's just rapidly reshaping life as we know it.
Which makes you ask, right, what does it actually mean to engineer life itself? And I mean, how is this incredible power going to redefine our future?
That's a really powerful way to frame it, yeah. What's truly profound here is just how far we've come. Shifting from just observing life to actively, well, editing its fundamental code. So today we're diving deep into this revolutionary world of genetic engineering, the precise manipulation of life's essential blueprints, DNA.
And we've got some great sources to guide us. You've shared some fascinating stuff, everything from academic courseware, really in-depth, to crash courses, and even cutting-edge research papers.
Yeah, good mix. We're ready to unpack the most, let's say, profound insights. We'll explore its origins, which are maybe more surprising than you think, demystify the tools that make it possible.
Really cutting-edge stuff.
Exactly. And maybe most importantly, consider how this science is already impacting humanity and, what it really means for the world of tomorrow.
Okay, so let's unpack this right from the beginning. To really get our heads around this revolution, we need a clear idea of the core concept. What is genetic engineering? Because it can sound a bit abstract, almost unreal.
It can, yeah. At its heart, genetic engineering. You might also hear it called genetic modification or gene manipulation or even recombinant DNA technology. It's fundamentally about deliberately altering an organism's genetic makeup.
Using technology specifically.
Right, using sophisticated technology. It's basically a set of precise tools that lets us change the cells of an organism, often by transferring specific genes, either within the same species or, and this is pretty amazing, even across entirely different species.
So it's not random chance, like evolution over millennia. This is targeted.
Very precise, very intentional change. Exactly.
No, and for you listening, it's probably helpful if we clarify some key terms. You'll hear batted around a lot in discussions about this.
Definitely. So any organism that's generated through genetic engineering is broadly called a genetically modified organism, or a GMO.
Right, GMO. That term pops up everywhere, often with a lot of baggage in public discussions.
It really does.
Yeah.
But the scientific definition is actually quite specific. It just means an organism whose genetic material has been altered using these genetic engineering techniques. Simple as that, technically speaking.
And there are some interesting distinctions even within that big GMO category. Like, for instance, if scientists take genetic material from another species and put it into an organism, what's that called?
That's called transgenic.
It's transgenic, okay.
Yeah, that glowing fish you mentioned at the start, the glowfish. Perfect example.
Ah, right.
It's a natural zebrafish, but it became the first genetically modified pet approved for sale in the U.S. back in 2003. And that was because scientists added genetic info from bioluminescent jellyfish to its DNA.
So literally a fish carrying jellyfish DNA, that's definitely transgenic.
Exactly. Clear case. Now, conversely, if the genetic material being introduced comes from the same species, or maybe from a species that could naturally breed with the host organism, then it's called cisgenic.
Cisgenic. Okay, so cis meaning same side, trans meaning across. Makes sense.
Precisely. And that distinction, it matters quite a bit, especially when you get into regulation and public perception. Cisgenic modifications are often seen as being a bit closer to traditional breeding, even though they still involve direct genetic manipulation.
Because it's staying within the species' natural gene pool, more or less.
Right. It maintains that closer relationship.
And sometimes it's not even about adding something new, is it? Sometimes it's about removing something.
Absolutely. If genetic engineering is used to remove a gene, or inactivate it, or as scientists often say, knock out a specific gene, the organism you get is called a knockout organism.
Knockout, got it.
And this is usually done in research. It's a powerful way to understand what a particular gene actually does. Or you can use it to model human diseases by disabling a gene that's known to cause a problem.
So you see what happens when that specific instruction in the blueprint is just missing.
Exactly. This helps figure out the function.
That's a really crucial point because it highlights how modern genetic engineering is just. It's a whole different ballgame compared to traditional breeding, isn't it?
Oh, completely. A real paradigm shift. I
And
mean, sure, humans have been altering genomes for millennia, absolutely, but it was through selective breeding.
like picking the corn with the biggest kernels or breeding wolves into all the dog breeds we have today?
Exactly, choosing the traits we liked. That process, turning wild wolves into domesticated dogs, started over 10,000 years ago. It's definitely a form of genetic alteration, but it works by leveraging the natural variation that's already there.
Right. You're selecting, not directly editing.
Precisely. But the modern genetic engineering we're talking about today, well, it's way beyond that. It's not just patiently picking the best traits generation after
It's
generation.
more like surgical.
It is. It's direct, precise manipulation of the DNA itself, right down to the molecular level. It's a fundamental shift in how we interact with life's code. We've gone from kind of nudging evolution along to actively, intentionally designing it.
That's a profound difference. And this level of precision, it's actually quite recent in the grand scheme of things.
Relatively speaking, yes. Although, interestingly, the term genetic engineering itself, it was actually coined way back in 1934.
No kidding. That early.
Yeah, by a Russian-born geneticist, Nikolai Timofey-Fusovsky. But the actual ability to do it, to perform these manipulations, that came much, much later. It really depended on foundational discoveries in molecular biology, understanding what DNA was and how it worked.
Okay, so the idea was there, but the tools weren't.
Exactly. And this is where the story gets really captivating, I think. The true dawn of modern genetic engineering, you could argue, started in 1972 with the groundbreaking work of Paul Berg.
Carl Berg, what did he do?
He created the very first recombinant DNA molecule,
Recombinant DNA, meaning.
meaning DNA made by combining genetic material from different sources. Specifically, he managed to link together DNA from two completely different viruses, the monkey virus, SV40, and a virus called lambda bacteriophage.
Wow. So mixing and matching virus DNA.
Essentially, yes. It was like the very first successful cut-and-paste operation in the genetic world. It proved that DNA from different origins could actually be joined together and still function. That was a huge conceptual and technical leap.
I can imagine. So that opened the door.
It really did. And the pace of innovation just exploded after that. Just 1 year later, in 1973, Herbert Boyer and Stanley Cohen took Berg's work a critical step further. They created the first transgenic organism.
The first actual GMO?
Basically, yes, a bacterium. They successfully inserted antibiotic resistance genes from one bacterium into a little circular piece of DNA called a plasmid in E. coli bacterium. And it worked. The E. coli became resistant to antibiotics it wasn't resistant to before.
Okay, so that wasn't just a linking DNA, that was making a functional change in a living thing.
Precisely. The birth of the first truly functional GMO. It showed immediately how powerful this recombinant DNA technology could be.
And the milestones kept coming.
They did. Building on that momentum, in 1974, Rudolf Janisch achieved another first. He created the world's first genetically modified animal, a transgenic mouse.
A GM mouse back in 74.
Yeah. He introduced foreign DNA into its early embryo, and the resulting mouse carried that altered genetic material and could even pass it on. These early breakthroughs, they weren't just, scientific curiosities. They immediately sparked huge discussions.
I bet. People must have realized pretty quickly how big this was.
Oh, yeah. Both the immense potential and also the profound ethical questions that came with his new power.
Did the scientists themselves worry about it?
They absolutely did.
Yeah.
The scientific community itself recognized the sheer magnitude of what was happening. Concerns about potential risks, how to use this responsibly. It led directly to the landmark Asilomar Conference in 1975.
Asilomar, I've heard of that.
It was pivotal. Leading scientists from all over the world gathered to discuss ethical guidelines for recombinant DNA research. They proposed a framework for responsible development, even including recommendations for government oversight.
So they tried to get ahead of the curve, ethically speaking.
They did, and that early self-regulation was absolutely crucial. It helped build trust and guide the field's development, making sure it proceeded, cautiously but responsibly.
And the practical applications weren't far behind, I assume.
Not at all. The first genetic engineering company, Genentech, was founded just a year later in 1976.
Genentech, still a huge name today.
Exactly. And by 1978, they'd achieved something truly revolutionary, producing genetically engineered human insulin.
Human insulin, that's massive for diabetes treatment.
Absolutely massive. Before this, insulin for diabetics had to be extracted from animal pancreases, usually pigs or cows. It was laborious, expensive, and sometimes caused allergic reactions in people.
Right.
Now, using genetic engineering, they could make bacteria produce actual human insulin, mass produce it. It became much more affordable, much more accessible, and safer. That product hit the market commercially by 1982, and it completely transformed diabetes management worldwide.
Just incredible. A direct medical benefit right out of the gate.
It was a huge success story. And that success in medicine started paving the way for other applications, especially in agriculture.
Moving from the pharmacy to the farm.
Right. By 1987, we saw the Ice- Pseudomonas syringae bacterium. This was genetically modified to protect crops from frost damage.
How did that work?
They removed a specific gene from the bacterium that actually promotes ice crystal formation on plants. So spray this modified bacteria on crops, and they become less susceptible to frost. This was one of the very first GMOs intentionally released into the environment, a big step from the lab to the field.
Was that controversial? Releasing a GMO into the environment sounds like it might raise some eyebrows.
Oh, it definitely did. It wasn't a smooth ride at all.
Yeah.
The initial field tests for the ice- bacterium were actually delayed for 4 years because of legal challenges from environmental groups. They were worried about potential ecological impacts, unintended consequences.
Understandable concern.
Yeah. And then, a pretty dramatic display of public feeling, when the trials finally got the go-ahead, the test fields themselves were actually attacked by activists the night before the test was supposed to happen.
Wow. So the public debate, the really passionate debate, has been there right from the start of commercialization.
Absolutely. It's not a recent phenomenon. But despite those challenges, the scientific and commercial momentum just kept building.
So the agricultural side pressed on.
It did. The first genetically engineered plants were being field tested by 1986. And then, in 1994, came the Flavsov tomato.
The what now?
Flavsov, Flavrsavr. It was engineered for longer shelf lives. They silenced a gene that causes tomatoes to soften as they ripen.
So a tomato that stays fresh longer because of genetic modification?
Exactly. And it became the first genetically modified food to be commercially approved in the U.S. That was another major milestone.
I bet. And now, where are we today with GM crops?
Well, it's gone way beyond just tomatoes. Today, it's not just a few niche things. There are actually 11 different transgenic crops being commercially grown in 25 countries around the world.
11 major crops. Like what?
Things like corn, soybeans, cotton, canola. Many engineered for traits like herbicide tolerance or insect resistance. It really shows the scale and how deeply integrated genetic engineering has become into our global food systems.
That widespread adoption really speaks volumes about the impact, especially in agriculture. And it brings us nicely to a really foundational story in this whole field, the Green Revolution.
Ah, yes, the Green Revolution. It's such a compelling story of how genetic understanding, even in its, let's say, earlier forms before direct DNA editing, could dramatically change human history.
And the key figure there was Norman Borlaug, right?
Absolutely. Norman Borlaug, an American geneticist and really a great humanitarian. Back in 1944, he set out on this mission to improve maize and wheat yields, especially wheat. It's the third most consumed cereal crop globally. Hugely important.
So what was the problem he was trying to solve?
Well, he developed these new wheat varieties that were high-yielding and resistant to common diseases, which was great. But they had long, thin stems.
Okay. Why was that a problem?
Because the stems were so flimsy, especially when the grain heads got heavy, they just fall over before harvest. It's a phenomenon called lodging.
Ah, so the plants literally couldn't support their own success.
Exactly. And this wasn't a minor issue. Lodging could reduce yields by up to 50%. Imagine losing half your crop just because the stems weren't strong enough.
Yeah.
Especially devastating in places facing food shortages.
Yeah, absolutely dire. So what did Borlaug do? He couldn't just tell the wheat to stand up straighter.
He couldn't, but he could use genetics. He and his team, leveraging their deep understanding of plant genetics, they did something really smart. They crossbred these high-yielding, disease-resistant but flimsy varieties with a specific Japanese dwarf wheat variety.
Or variety Y.
Because it was naturally short and sturdy. By crossbreeding, they successfully introduced this non-falling over trait, the dwarfing trait, into the high-yielding, disease-resistant wheat. It was a brilliant strategic intervention using natural genetic variation to solve a critical agricultural problem.
So basically, short, sturdy, high-yielding, disease-resistant wheat, the whole package.
That was the goal, and they achieved it.
Right.
And the impact, Well, it was nothing short of monumental.
How so?
These new semi-dwarf, high-yielding wheat varieties were introduced, starting around 1962, into countries like India and Pakistan, places that were really struggling with food security.
Yeah.
And they essentially doubled crop yields in that region over the next decade. Doubled.
Wow. Doubled yields. The impact on food availability must have been
It
enormous.
is incredible. This achievement became known worldwide as the Green Revolution, and it's widely credited with rescuing an estimated 300 million people from starvation. 300 million.
That's staggering. It really puts the power of applied genetics into perspective.
It really does. And for his tireless work and his profound global impact, Borlaug was awarded the Nobel Peace Prize in 1970. Quite rightly so. It's just this extraordinary example of how understanding genetics, even before modern direct DNA editing, could tackle a massive global crisis and fundamentally improve human well-being.
It truly highlights that transformative power, and you can see how that success must have paved the way for, more advanced genetic engineering later on.
Absolutely, it built confidence and demonstrated the potential. As we mentioned, the Ice-minus bacterium came along by 87, engineered plants were tested in 86.
Right, and the flavsavor tomato proved in 94.
Exactly. The commercial momentum kept building, driven by these kinds of clear potential benefits. And today, as we said, 11 major transgenic crops grown across 25 countries, herbicide tolerance, insect resistance, things that directly help farmers and potentially increase yields.
So the key insight there is really how those early successes, even using more traditional breeding combined with genetic knowledge, laid the groundwork. It set the stage for this whole new era of really precise genetic intervention we see today, tackling big challenges like food security head-on.
Precisely.
Okay, so we've talked about what genetic engineering is, we've looked at its history, the Green Revolution, but now the big question. How does it actually work? How do scientists engineer life's code? This is where we need to get into the nitty-gritty, the actual mechanics.
Right. Let's deconstruct the deep dive into the process itself. It all comes down to understanding DNA.
The blueprint of life.
Exactly. Think of DNA as the ultimate instruction manual for every living thing on Earth. This incredible molecule is made of just 4 fundamental chemical letters or bases: adenine, thymine, guanine, and cytosine, A, T, G, C.
The 4 letters of the genetic alphabet.
That's right. And the specific sequence of these 4 bases forms the biological code. It dictates everything, a cell's structure, how it operates, everything. Segments of this long sequence make up genes.
And the genes determine the traits.
Precisely. Genes determine an organism's characteristics, why a tiger has stripes and a jaguar has spots, why your hair is the color it is, why sunflowers turn towards the sun. It's all written in that fundamental code.
So genetic modification is about editing that code. How does that process actually happen? Is it like typing on a keyboard?
Not quite that simple, unfortunately. It's more like a complex, multi-step dance, a really choreographed sequence. And it starts, perhaps, obviously, with finding the right instruction you want to work with.
Choosing the right gene.
Exactly. Choosing the specific gene, the piece of the instruction manual that you want to add or maybe remove or perhaps just change slightly.
How do they find the right one? Genomes are huge and complicated.
They are, but modern tools make it much easier than it used to be. Things like advanced genomic scanning techniques, microarrays, transcriptomics, whole genome sequencing allow scientists to quickly identify specific genes across an organism's entire genetic blueprint.
So they can search the entire library, so to speak.
In a way, yes. But sometimes, it's just pure luck, pure serendipity. Like the discovery of the famous Roundup Ready gene.
Roundup Ready, that's the herbicide resistance, right?
That's the one. Scientists basically just noticed a bacterium that was happily thriving in soil contaminated with the herbicide Roundup. They investigated, found the gene responsible for that resistance, and realized they could potentially transfer that resistance into crop plants.
So sometimes it's detective work, sometimes it's high-tech scanning.
Exactly. Once you've identified your target gene, the next step is to isolate it and make lots and lots of copies.
Why copies?
Well, you need enough of the gene to work with for analysis, for modification, and for the insertion process later.
Right.
Isolating it often involves using those molecular scissors we talked about, restriction enzymes.
Right, the enzymes that cut DNA at specific spots.
Precisely. They recognize particular DNA sequences and make a cut.
Yeah.
Or, more commonly nowadays for making many copies, scientists use a technique called polymerase chain reaction, PCR.
PCR. That sounds familiar. It's used in more than just genetic engineering, isn't it?
Oh, absolutely. PCR is truly a foundational technology in modern biology. It's fascinating because it's become this incredibly powerful and frankly, ubiquitous diagnostic tool that has reshaped entire fields.
Like what?
Well, for instance, it's used constantly in diagnosing diseases, bacterial, viral, things like tuberculosis, hepatitis, HIV. It works by taking tiny amounts of the pathogen's DNA or RNA, maybe from a blood sample, and amplifying it, making millions or billions of copies very quickly so it becomes detectable.
Okay, so for medical diagnosis.
Definitely. And in forensics, medical legal cases, PCR can amplify minute DNA samples from a single hair follicle, a tiny drop of blood, to create a unique genetic fingerprint. This is used for RFLT analysis, helping to identify individuals in criminal investigations or paternity cases.
Right, DNA fingerprinting.
Exactly. It's also critical for diagnosing inherited genetic disorders like thalassemia or cystic fibrosis, often allowing for very early detection, sometimes even before birth.
That's huge for families.
It is. And in cancer detection, too. It helps monitor for residual abnormal cells after treatment or identify specific gene mutations, like mutations in the p53 gene, which are linked to many cancers.
So it's a really versatile tool.
Incredibly versatile. Even in paleontology, scientists use PCR to amplify tiny fragments of ancient DNA recovered from fossils, allowing them to compare genetic sequences in extinct organisms like Neanderthals with living ones.
Wow, from diagnosing HIV to studying Neanderthals, that's quite a range.
It really is. The profound insight here is how this one molecular technique, PCR, has become this kind of invisible backbone, supporting everything from forensic science to diagnosing global pandemics like COVID-19, and even understanding our evolutionary past. It just shows the incredible power of being able to target and manipulate DNA.
Okay, so back to the genetic engineering process. You found your gene, you've used PCR to make millions of copies. What's next? You need to store these genes.
Well, yes, for ongoing work. That's where gene libraries come in. Think of them as vast, organized collections of cloned DNA fragments from all sorts of organisms, including humans.
Like a library, but for genes instead of books.
Exactly. They're like immense, searchable databases of genetic information, providing researchers with an almost unlimited supply of specific genes for their projects.
Okay, so you've got your gene, isolated and copied. Now you need to actually put it into the target organism. Is it just a matter of injecting it?
Not quite. Before insertion, the gene usually needs to be prepared. It's typically combined with other essential genetic elements within a small piece of DNA, often a plasmid.
Other elements, like what?
You need things like a promoter region. Think of that at the on switch. It tells the cell when and where to actually read and use the new gene. You don't necessarily want it active all the time in every cell.
So you can control its expression.
Exactly. And you also need a terminator region, which signals the end of the gene's instructions, telling the cellular machinery, okay, stop reading here.
Like punctuation for the genetic sentence.
Good analogy, yes. And very often, you also include something called a selectable marker. This is usually a gene that confers resistance to a specific antibiotic.
Why antibiotic resistance?
It acts like a flag. After you try to insert the new DNA package, you can treat the cells with that antibiotic. Only the cells that successfully took up the new DNA, which includes the resistance gene, will survive. It makes it easy to identify and select the cells that have actually been successfully modified.
Clever. A way to filter out the failures.
Precisely. It's a crucial step for efficiency.
Okay, so the gene package is ready. Now for the insertion, the actual delivery. How does that work? Does it vary depending on the organism?
It varies quite significantly, yes. Let's take bacteria first. Some types of bacteria naturally just slurp up foreign DNA from their environment. It's a process called transformation.
They just take it in?
Some do, yes. Others need a bit of persuasion. You can induce them to take up DNA by applying some kind of stress, like a brief electric shock, that's called electroporation, or a sudden temperature change called heat shock. This makes their cell membranes temporarily leaky, allowing the plasmid DNA carrying the new gene to get inside.
Okay, so you can shock bacteria into taking new DNA. What about animals? Can't really shock a mouse into taking DNA.
Haha, no, not quite like that. For animal cells, the DNA is usually introduced more directly. One common method is microinjection.
Microinjection. Sounds delicate.
It is. You use an incredibly fine needle under a microscope to inject the genetic material directly into the nucleus of the target cell, often an egg cell or an early embryo cell.
Wow. Directly into the nucleus.
Yep. Another very common and highly effective method, especially for therapeutic applications, is using viral vectors.
Using viruses. Aren't they usually the bad guys?
Normally, yes. But here, scientists cleverly hijack them. They take a virus, strip out its harmful disease-causing genes, and replace them with the therapeutic gene they want to deliver. The modified virus then acts like a highly efficient delivery truck, using its natural ability to infect cells to carry the beneficial gene into the host's cells.
So you turn the virus into a delivery system for good genes. That's ingenious.
It's a very powerful technique, widely used in gene therapy research.
Okay, bacteria, animals. What about plants? How do you get DNA into plant cells? They have tough cell walls.
They do, which presents a different challenge. Several clever methods are used. One very common approach uses a bacterium called Agrobacterium tumefaciens.
Agrobacterium. Sounds familiar. Doesn't that cause tumors in plants?
Naturally, yes, it causes crown gall disease. But scientists have figured out how to disarm it, removing the tumor-causing genes and replacing them with the genes they want to insert into the plant. They basically reprogram the bacterium to deliver desired genes instead of disease genes.
So again, hijacking a natural system.
Exactly. Another method is called biolistics, which sounds pretty dramatic.
Biolistics.
Yeah, it's literally a gene gun.
A gene gun.
Seriously, it fires tiny particles, usually gold or tungsten, that have been coated with the DNA you want to insert directly into young plant cells or tissues. It physically blasts the DNA through the cell wall.
You shoot DNA into the plant cells? Okay, that's definitely science fiction made real.
It kind of is. And then, similar to bacteria, you can also use electroporation on plant cells, that electric shock method, to make the cell membranes temporarily permeable to the DNA.
So lots of different methods depending on what you're working with. After all that, after the insertion, what happens? You have one modified cell.
Right. And now you need to regenerate a whole organism from that single modified cell or group of cells. For plants, this is typically done using tissue culture.
Tissue culture?
Yeah, you take the modified cells and grow them on a special nutrient medium in the lab. Under the right conditions, you can actually regenerate an entire plant from just a few cells. It's quite remarkable.
Wow, and for animals?
For animals, it's often more complex. It might involve using embryonic stem cells, which can develop into any cell type, or other advanced reproductive techniques like cloning or transferring the modified nucleus into an egg cell.
Okay, so you grow your modified plant or animal, is that it? How do you know for sure it worked properly?
Ah, good question. Rigorous testing is absolutely essential at the end. You need confirmation. Techniques like PCR, which we discussed, can confirm if the new gene is actually present in the organism's DNA. DNA sequencing can read the exact genetic code to make sure the gene was inserted correctly and hasn't been damaged.
So you check if the gene is there and intact.
Exactly. But just being there isn't enough. You also need to know if it's working, if it's being expressed.
Meaning, is it actually doing the job it was put there to do?
Precisely. Is it producing the desired protein or RNA molecule? So scientists use other sophisticated tests looking at RNA levels, protein levels, to confirm that the new gene is actually being switched on and functioning as intended, leading to the desired trait, whether that's glowing in the dark or resisting a disease.
It's a really complex, multi-stage process requiring incredible precision.
It absolutely is. And it showcases the level of control that modern genetic engineering offers, a level of detail and specificity that was just unimaginable with traditional breeding. While these foundational methods are still used and constantly being refined, the last decade or so has seen the emergence of truly revolutionary tools that have just completely changed the landscape again.
Ah, okay. This is where we get into the really cutting-edge stuff that you hear about in the news more often now.
Exactly. Tools that have opened doors we once only dreamed of, particularly in areas like understanding the brain or making gene editing much easier.
Right. You mentioned two big ones earlier, optogenetics and CRISPR. Let's start with optogenetics. That sounds like light genetics.
Sort of. It's a breakthrough technique that involves modifying specific cells, very often brain cells, neurons, to make them exquisitely sensitive to light.
Making brain cells light sensitive, why would you want to do that?
It sounds strange, I know. But it has profound applications for understanding how the brain works and potentially treating neurological disorders. Here's the basic idea. Brain cells have these tiny little gatekeepers on their surfaces called ion channel receptors.
Okay, gatekeepers.
Normally, specific chemicals in the brain, neurotransmitters, bind to these receptors. That binding opens or closes the gates, letting ions flow in or out, which causes the neuron to activate or deactivate to fire or not fire.
So chemicals control the brain cell's activity.
Right. Now, here's the clever part in optogenetics. Scientists use those viral vectors we talked about, the modified harmless viruses.
The delivery trucks?
Exactly. They use them to deliver DNA into specific neurons. But this DNA contains the instructions for building light-sensitive ion channels, often borrowed from algae or other microbes that naturally respond to light.
So you're adding light-activated gates to the brain cells.
Precisely. You essentially rewire these gatekeepers so that light becomes the key to opening or closing them.
Whoa, so instead of a chemical signal, you can use, what, like a tiny laser beam?
Exactly. You can implant tiny optical fibers into the brain region you're studying. Then, by shining precise flashes of light of specific colors, you can turn specific types of neurons on or off, literally at the speed of light, with incredible temporal precision.
That's unbelievable. You can control brain circuits with light switches.
Essentially, yes. And this offers an incredibly powerful way to figure out what different brain circuits actually do. You can activate a specific set of cells and see what behavior results, or silence them and see what function is lost.
So you can map brain function with pinpoint accuracy?
Right. It's building massively on insights we got from, say, studying the motor cortex in mice or trying to unravel complex sleep cycles in fruit flies. It allows researchers to precisely control neural activity and observe the direct effects, giving us unparalleled insights into brain function and dysfunction.
And the potential for therapies.
Huge potential down the line. Imagine being able to precisely stimulate or inhibit malfunctioning brain circuits in conditions like Parkinson's disease or epilepsy or even depression, using light instead of drugs or crude electrical stimulation.
That's incredible. The key insight there really is that ability to control biological functions with an external trigger like light. It just opens up whole new ways of investigating complex systems.
It really does. It's revolutionized neuroscience research.
Okay, so that's optogenetics. Mind-blowing. What about the other big one? CRISPR. You hear that name everywhere now.
CRISPR, yes. Short for clustered, regularly interspaced, short palindromic repeats, which is quite a mouthful.
Definitely. What does it actually mean?
Well, the name refers to a pattern found in bacterial DNA. But what CRISPR is, in practice, is an absolute game changer in genetic engineering. It's often described as life's molecular scissors, but it's even more versatile than that.
How so? What makes it different?
Several things. Compared to older gene editing methods, CRISPR is remarkably easier to customize and much more efficient. Crucially, it can be used to both remove specific genes or parts of genes from a cell and also to add new genetic information at a precise location.
So it's like a biological find and replace tool for DNA.
That's a great analogy, yes. Or like being able to literally cut and paste DNA sequences with unprecedented ease and precision.
Okay, so where did this amazing tool come from? Did scientists invent it from scratch?
Actually, no. And this is the really brilliant part. CRISPR isn't something humans invented. It's something we discovered and adapted. It's based on bacteria's own natural ancient defense system against viruses.
Bacteria have immune systems.
They certainly do. Bacteria are constantly under attack from viruses called bacteriophages.
Oh.
So over millions of years, they evolved this incredibly clever way to fight back.
How does it work?
Okay, so when a bacterium survives a viral infection, special enzymes inside the bacterium actually snip out small pieces of the invading virus's DNA.
They take a sample of the enemy DNA.
Exactly. And then they integrate these snippets into their own genome, into specific regions called CRISPR arrays. These stored viral snippets act like a genetic mugshot library, a memory of past attackers.
A most wanted list for viruses.
Kind of, yeah. Then, if that same type of virus tries to infect the bacterium again later, the bacterium quickly transcribes those stored viral mugshots into small RNA molecules. These are called guide RNAs.
Guide RNA.
These guide RNAs then team up with a special DNA-cutting enzyme, the most famous one being called Cas9. Think of Cas9 as the molecular scissors or the attack dog.
So the guide RNA tells the Cas9 where to go.
Precisely. The guide RNA, carrying the viral mugshot, directs the Cas9 enzyme to scan the DNA inside the cell. If it encounters any DNA that matches the mugshot sequence, meaning if it finds the invading virus's DNA...
It recognizes the enemy.
Right. And when it finds that match, the Cas9 enzyme does its job. It cuts the viral DNA, chopping it up and neutralizing the threat, preventing the virus from replicating and harming the bacterium.
Wow. That's a really sophisticated defense system. So how did scientists turn that into a gene editing tool for, say, human cells?
That was the genius leap. Researchers, particularly Jennifer Doudna and Emmanuel Charpentier, who won the Nobel Prize for this, realized they could hijack this bacterial system. They figured out that they could create their own custom guide RNAs in the lab.
Guide RNAs that could match any DNA sequence they wanted to target, not just viral DNA.
Exactly. They could design a guide RNA to match a specific gene in a plant or an animal or even a human cell. Then if you introduce that custom guide RNA along with the Cas9 enzyme into a cell,
The guide RNA leads the Cas9 scissors right to that specific gene you targeted.
Cas9 cuts the DNA precisely at that spot.
Okay, so you can cut DNA wherever you want. How does that help you edit it?
Well, when DNA gets cut like that, the cell has natural repair mechanisms that kick in to fix the break. Sometimes the repair process is a bit sloppy and introduces small errors, which can effectively disable or knock out the gene you targeted.
Ah, so you can use it to remove gene function that way.
Yes. Or, and this is where it gets really powerful, you can also provide the cell with a new piece of DNA at the same time, a template sequence.
Oh, replacement part?
Kind of. The cell's repair machinery can then use that template to fix the break, essentially pasting the new sequence into the DNA at the cut site. This allows you to precisely change the existing gene sequence or even insert a whole new gene.
So you can cut, disable, replace, or insert. That's incredibly versatile.
It really is. That's why CRISPR has just revolutionized genetic engineering. It's made gene editing so much faster, cheaper, and more accessible than previous technologies,
The potential sounds enormous.
especially for diseases.
Oh, the theoretical potential is just staggering. Think about curing inherited genetic diseases, conditions like cystic fibrosis, sickle cell disease, Huntington's disease, certain types of muscular dystrophy.
Diseases caused by specific mutations in single genes.
Exactly. With CRISPR, the idea is you could potentially go into the relevant cells, find that faulty gene sequence, cut it out, and replace it with the correct healthy version. Correcting the typo in the genetic code, basically.
That's the dream. Imagine eliminating that inherited suffering. Or think about cancer, maybe using CRISPR to re-engineer a patient's own immune cells to better recognize and attack cancer cells. The possibilities seem almost endless.
It really sounds like a revolution in medicine waiting to happen.
Hmm.
But there have to be caveats, right? It sounds almost too good to be true.
There are definitely caveats, and it's crucial we understand them. While CRISPR offers incredible precision compared to older methods, it's still not absolutely perfect, especially in its current forms.
What can go wrong?
The biggest concern is off-target effects,
Meaning,
meaning the Cas9 enzyme sometimes cuts the DNA in unintended places, locations that are similar but not identical to the target sequence.
so it cuts the wrong gene or somewhere else entirely.
It can happen, yes. And these off-target cuts can have unpredictable consequences. They could potentially disrupt other important genes or cause mutations that might even lead to other problems, like cancer.
So trying to fix one problem could accidentally create another one.
That's the risk. Scientists are working incredibly hard to improve the specificity and accuracy of CRISPR systems to minimize these off-target edits. But it's still an active area of research and a major safety concern, especially for therapeutic applications in humans.
Yeah, you'd need to be incredibly sure it's safe before editing human DNA therapeutically.
Absolutely. Changing DNA, even with the best tools, isn't something to be taken lightly. If you make an incorrect edit, especially in the wrong cells, you could potentially cause the very kinds of genetic problems you're trying to cure.
So despite the huge excitement, there's still a way to go before we're routinely and safely genetically engineering humans at the DNA level.
Definitely. Especially when we start talking about inheritable changes, editing eggs, sperm, or embryos,
Right.
which would be passed down to all future generations. That raises even bigger safety and ethical questions. The key insight here is that CRISPR is this quantum leap in gene editing power and accessibility, but that very power demands extreme caution, ongoing research to ensure safety, and really deep societal discussion about how we use it ethically.
Which makes sense. But even with those limitations and the need for caution, the applications of genetic engineering using CRISPR and other tools are already reshaping our world in profound ways, right?
Yeah.
Touching almost every aspect of our lives.
They absolutely are. Let's dive into some of those impacts, starting with medicine, where the transformations have been just incredible.
Okay, medicine first. We already mentioned insulin.
Right. Insulin was one of the first big successes. But genetic engineering has profoundly revolutionized the production of many other pharmaceutical drugs, too. Before this tech, getting human proteins for medical use was often incredibly difficult, really expensive, or relied on harvesting from human or animal sources, which had limitations in supply and sometimes caused allergic reactions.
So what else besides insulin?
Well, human growth hormone, vital for treating growth disorders. Follicle-stimulating hormone used in fertility treatments. Human albumin, used in various medical situations. And maybe most significantly today, monoclonal antibodies.
Monoclonal antibodies, they're used for a lot of things now, right?
Mm,
Cancer, autoimmune diseases.
exactly. They are incredibly important targeted therapies. And now, thanks to genetic engineering, they can be mass-produced efficiently and safely in genetically engineered cells, often grown in large bioreactors. This makes these life-saving treatments much more widely available and generally safer than older methods. It reduces costs, increases supply, ensures purity. It's been a huge win for medicine.
That's fantastic. What about research? How has it changed medical research?
Massively, particularly through the use of animal models. Genetically modified mice, especially, have become absolutely indispensable tools for researchers.
Why mice?
Well, they're mammals, relatively easy to breed and handle, and their genetics are surprisingly similar to humans in many ways. So, scientists can engineer mice to mimic human diseases with remarkable accuracy.
So you can create a mouse model for, say, Alzheimer's.
Absolutely. Or various forms of cancer. The oncomouse, engineered to be susceptible to cancer, was one of the very first patented GM animals back in the 80s. Models for obesity, heart disease, diabetes, arthritis, Parkinson's, Huntington's, even complex conditions like substance abuse or anxiety. You name it, there's probably a GM mouse model for it.
And what do researchers do with these mice?
They allow researchers to study how these diseases develop and progress in a controlled living system. They can unravel the complex biological pathways involved, and, crucially, they can rigorously test potential new drugs and therapies in these models before they ever move to human clinical trials. It's an essential step for safety and efficacy testing.
Absolutely vital. And then, of course, there's the ultimate promise, the most ambitious application, gene therapy directly in humans.
By actually fixing faulty genes in people.
Exactly. The idea is to replace defective genes with functional ones to cure or at least treat genetic disorders at their root cause.
Is this actually happening now? Or is it still mostly theoretical? It's happening. Clinical research is actively ongoing for a whole range of devastating diseases. Things like X-linked SCID, severe combined immunodeficiency, sometimes called bubble boy disease, where children are born with essentially no immune system. Gene therapy has shown real success there. Also, research into Parkinson's, hemophilia, certain types of blindness.
So real progress is being made.
Definitely. In 2012, the very first gene therapy treatment, called Glibera, for a rare inherited metabolic disorder, was actually approved for use in Europe. That was a huge milestone.
The first approved gene therapy.
Yes. And then in 2015, there was that truly remarkable case you might have heard about a young boy in Germany suffering from a horrific, life-threatening skin disease called epidermolysis bullosa, where the skin just blisters and tears
Oh,
constantly.
I think I remember reading about that. His skin was falling off.
Essentially, yes. It was incredibly severe. Doctors took some of his skin cells, used a viral vector to deliver a corrected version of the faulty gene into those cells in the lab, grew sheets of this genetically corrected skin, and then grafted it back onto him.
They gave him new, healthy skin.
Over 80% of his body surface. And it worked. It essentially saved his life and dramatically improved his quality of life. It was just a stunning demonstration of the power of somatic gene therapy.
Somatic. What does that mean?
Somatic gene therapy means it targets the non-reproductive cells of the body, like skin cells, blood cells, lung cells. So the changes are made only in that individual and are not passed on to their children. It doesn't affect the germline.
Okay. So it avoids those ethical concerns about inheritable changes.
Exactly. That's why most current gene therapy research focuses on somatic cells.
It really makes you wonder, though, doesn't it? As this technology gets better and better,
Yeah.
what if, What if we could actually eliminate all genetic diseases before birth? Maybe by correcting errors in the embryo stage, assuming we could do it safely and ethically?
That's the big what ifs, isn't it?
Hmm.
Imagine a future where conditions like cystic fibrosis or Huntington's disease or sickle cell anemia are just gone.
Hmm.
Things of the past, eradicated through genetic precision.
It's hard to even comprehend. Or what about other medical challenges? Like organ transplants. There's a huge shortage.
A critical shortage, yes. But
Yeah.
what if? What if we could engineer pigs, for example, to grow organs, hearts, kidneys, livers that are fully compatible with the human immune system, eventually creating an unlimited supply of organs on demand?
Are people actually working on that? Pig organs for humans?
Oh, yes. Scientists are intensely working on it right now, using CRISPR and other tools to make multiple genetic modifications in pigs to knock out pig genes that trigger rejection in humans and even add human genes to improve compatibility. There have already been some experimental transplants of pig kidneys and hearts into brain-dead human recipients with promising initial results. It's called xenotransplantation.
Xenotransplantation, wow! That could solve the organ shortage crisis.
Potentially, yes. It's still facing hurdles like long-term rejection and the risk of transmitting animal viruses, but the progress is rapid.
And what about infectious diseases, like malaria?
Another huge area of potential, consider the concept of a gene drive.
Gene drive, what's that?
It's a genetic engineering technique designed to spread a particular genetic modification rapidly through an entire population, much faster than normal inheritance would allow.
How does that work?
Basically, you engineer the gene you want to spread, say a gene conferring malaria resistance in mosquitoes, so that it also carries the instructions for CRISPR-Cas9 targeted to the original version of that gene. When an engineered mosquito mates with a wild one, the CRISPR system in the offspring essentially cuts the original gene inherited from the wild parent and replaces it with the engineered version.
So it forces the inheritance of the modified gene?
Pretty much, yeah. Almost all offspring inherit the modified gene, not just 50%. So the trait can spread incredibly quickly through the whole population over just a few generations.
And people are trying this for malaria.
Yes. Scientists are actively developing gene drives to make mosquitoes unable to transmit the malaria parasite, or even to crash mosquito populations entirely. The idea is you could release a relatively small number of engineered mosquitoes, and the gene drive would spread that anti-malaria trait through the wild population, potentially eradicating the disease in a region.
That's incredible, but also sounds potentially risky if something went wrong.
Extremely risky. Gene drives are very powerful and potentially irreversible in the wild. There's intense debate about the ecological consequences and the need for safeguards before any environmental release. But the potential benefit for diseases like malaria, which kills hundreds of thousands of people every year, mostly children, it's hard to ignore.
These are truly transformative possibilities, not just tweaking things, but potentially solving massive global health problems. These aren't distant sci-fi dreams anymore. They're active areas of research pushing the boundaries right now.
Absolutely. And beyond human health, genetic engineering is making equally transformative strides in agriculture, enhancing food security, improving sustainability, especially critical in a world facing climate change and a rapidly growing population.
Right. We touched on the Green Revolution, but modern genetic engineering in agriculture offers even more precision, doesn't it?
It really does. It offers a level of precision and speed that conventional plant breeding, even with decades of work, simply can't match.
How so?
Well, as we discussed, genetic engineering uniquely allows for the interchange of genetic material across species boundaries. Introducing a gene from a bacterium into a plant to confer insect resistance, for instance. Traditional crossbreeding could never do that.
Right, you can only cross related species.
Exactly. And furthermore, it allows for the precise introduction of new genes into crops that are really difficult to improve through conventional breeding because of how they reproduce. Think about crops that are propagated vegetatively, like bananas, cassava, potatoes. They don't rely on seeds in the same way, making traditional crossbreeding very slow and cumbersome. Genetic engineering can bypass those limitations.
So you can improve crops that were hard to improve before. Have we seen many successes come to market?
We have. A remarkable range of approved crops are out there now, directly tackling major agricultural headaches. We mentioned herbicide tolerance and insect resistance in crops like corn and soy. But there are also varieties of squash, papaya, and even potatoes that have been engineered to be resistant to devastating viruses or fungal blights.
Like the potato blight that caused the Irish famine. Could GE prevent that?
Well, scientists have developed potato varieties resistant to the specific fungus that causes late blight. Yes. These engineered varieties can significantly reduce crop losses and dramatically cut down on the need for fungicide sprays, which is better for the environment and farmers' costs.
Papaya, too.
Yes, the papaya industry in Hawaii was nearly wiped out by the ring spot virus in the 1990s. Genetically engineered, virus-resistant papaya varieties essentially saved the industry there. Even common beans, a staple food in many parts of the world, have been engineered to resist the bean golden mosaic virus, which can just devastate harvests in tropical regions.
So it's really about protecting crops from diseases and pests. Are the strategies getting more sophisticated?
Incredibly sophisticated, yes.
Yeah.
It's not just about inserting one resistance gene anymore. For example, plants are now being engineered to produce their own antimicrobial peptides, small proteins that directly attack bacteria or fungi.
So the plant makes its own medicine.
Sort of, yeah. Or they can be engineered to exploit their own natural defense system called RNA interference, or RNAi.
RNAi. How does that work for immunity?
The plant can be engineered to produce small RNA molecules that specifically match essential genes in an invading virus. These small RNAs then guide cellular machinery to find and destroy the viral RNA, effectively silencing the virus and giving the plant immunity. It's like a targeted antiviral system built into the plant.
Clever.
Very. And researchers are even developing what are called immune receptors proteins, either natural ones enhanced or completely engineered ones, that sit on the plant cell surface and can recognize a much broader range of different pathogens. When they detect a pathogen, they trigger a strong, rapid defense response in the plant.
Like giving the plant a more advanced immune system.
And that's a good way to think about it.
Hmm.
And another really cutting-edge approach is to actually modify the plant's own genes, specifically targeting what are called susceptibility genes.
Susceptibility genes. Genes that make the plant vulnerable.
Exactly. These are genes that pathogens have evolved to hijack or exploit for their own benefit during infection. For instance, many pathogens manipulate the plant's sugar transport genes, like the sweet family genes, to steal sugars from the plant cells.
So the pathogens trick the plant into feeding them.
Right. So if scientists use gene editing, like CRISPR, to slightly modify those sweet genes in the plant, they can make it much harder for the pathogen to access the sugars, thereby enhancing the plant's resistance. This has shown great promise against bacterial blight in rice, a major global crop.
So instead of adding a defense gene, you're removing a vulnerability.
Precisely.
Hmm.
Similarly, knocking out or modifying genes like EIF4E in crops like cucumber has been shown to create broad resistance to a range of different viruses because many viruses need that specific plant protein to replicate. By tweaking the plant's own machinery, you can make it inherently less hospitable to disease.
That sounds like a really smart, targeted approach. Is GE in agriculture only about disease and pest resistance?
Not at all. That's a major focus. But GM crops are also being developed for lots of other traits. Increased yield, obviously growing more food on the same amount of land, is crucial for feeding a growing population.
Higher productivity.
Right. Also, enhanced tolerance to environmental stresses. Think about crops that can better withstand high salt levels in the soil, which is a growing problem in irrigated areas. Or crops tolerant to extreme cold, or perhaps most importantly, drought. Making crops more resilient to drought could be hugely important for adapting agriculture to climate change.
Making farming possible in tougher conditions.
Exactly. And beyond just quantity and resilience, genetic engineering is also improving nutritional quality.
Like golden rice, the rice with vitamin A.
Golden rice is probably the most famous example, yes. Engineered to produce beta-carotene, a precursor to vitamin A, to combat deficiency in regions where rice is a staple. But there are other examples too, like soybeans engineered to produce healthier cooking oils with less saturated fat and more beneficial fatty acids.
So improving the nutritional profile of food.
Right. Or even tailoring crops for industrial uses. There was a GM potato developed in Europe called Amflora. It was engineered to produce almost pure amylopectin starch, which is much more useful for industrial applications like papermaking and adhesives compared to the normal mix of starches in potatoes.
So engineering crops for specific industrial outputs too.
Yes. And that brings us to another fascinating and growing area, something called farming.
Farming, like farming pharmaceuticals.
Exactly. It's a portmanteau of farming and pharmaceuticals.
Okay.
This is where crops, or sometimes animals, are genetically engineered to act as living bioreactors.
Bioreactors. To produce what?
To produce valuable pharmaceutical products, vaccines, therapeutic antibodies, enzymes, other complex protein drugs.
So instead of making these drugs in expensive factory facilities, you grow them in a field.
That's the idea, yes. Or in an animal. Imagine, for instance, cows or goats engineered to express a complex human medicine in their milk. The milk can then be collected and the drug purified relatively easily.
Growing medicine in milk, is that actually happening?
It is. This method has already seen FDA approval for a couple of drugs. For example, a drug called Atrin, an anticoagulant protein, was approved back in 2009 and is produced in the milk of genetically engineered goats. It offers a potentially scalable and cost-effective way to produce certain complex biological drugs.
That's incredible. So plants and animals as living drug factories.
It highlights the incredible versatility of genetic engineering. It's not just about food, it's providing unique, precise, and often much faster solutions to challenges that once seemed almost insurmountable, from improving crop resilience against climate change to revolutionizing how we manufacture life-saving medicines.
The applications just keep expanding. Beyond health and food, are there other areas where genetic engineering is making a mark, maybe less obvious ones?
Oh, definitely. The applications are incredibly diverse, sometimes quite surprising, touching industries, conservation, even art.
Okay, like what in industry?
Well, we're seeing bacteria being engineered to become much more efficient at producing sustainable biofuels like ethanol or butanol from plant waste or algae. This could be really important for helping us transition away from fossil fuels.
Using microbes to make fuel.
Right. Or using engineered microbes for environmental cleanup. There are bacteria being developed that can efficiently break down toxic pollutants, like those found in massive oil spills, literally eating the oil,
Hmm.
or microbes designed to detect harmful contaminants, like arsenic, in drinking water supplies, acting as tiny biological sensors.
Bioremediation and biosensors.
Exactly. There are even bacteria engineered to help extract valuable heavy metals from mining waste or contaminated water, concentrating them so they can be recovered. It's part of a field called biomining or bioremediation.
Using biology to clean up our messes or extract resources.
Precisely. And in a really surprising twist, researchers at MIT actually used a genetically modified virus, a harmless bacteriophage, as a kind of biological scaffold to assemble the components of a lithium-ion battery cathode.
A virus building a battery.
Essentially, yes. The virus was engineered to grab onto certain materials and arrange them in a precise structure at the nanoscale, leading to a battery that was potentially more powerful and could be assembled in a more environmentally friendly way at room temperature.
That's wild. The industrial possibilities seem huge, offering potentially greener, more sustainable ways to do things.
They really are vast.
Yeah.
And it extends into conservation biology too, although many of these applications are still more theoretical or in very early stages.
Conservation? How could genetic engineering help save species?
Well, imagine the potential to control invasive species, perhaps by engineering them with a gene drive, like we discussed for mosquitoes, but designed to reduce their reproductive capabilities, causing a population crash in a targeted, maybe more humane way than traditional culling or poisoning.
A genetic solution to invasive species. Controversial, I imagine.
Highly controversial, yes, because of the risks of unintended spread, but is being explored. Or think about vaccinating endangered animals against devastating diseases. Maybe not by catching and injecting them all, but by engineering a transmissible vaccine, or even engineering the animals themselves for inherent genetic immunity to specific threats.
Genetic immunization for wildlife.
Potentially.
Mhm.
And what about trees? Huge forests are threatened by newly emerging pathogens, often exacerbated by climate change. Could we engineer trees, perhaps important keystone species, to resist these pathogens? It's sometimes called facilitated adaptation, using genetic tools to help species cope with rapid environmental change that natural evolution can't keep up with.
Helping nature adapt faster using GE. That's a fascinating idea.
It is, again, lots of ecological questions, but it's being seriously discussed as a potential tool for conservation in the face of unprecedented change. It's really about leveraging biology itself to try and protect and restore our natural world.
Okay, industry conservation. You mentioned art. How does genetic engineering fit into art?
Uh-huh. Well, maybe art is stretching it sometimes, but certainly novelty and aesthetics. It demonstrates the sheer breadth of possibilities. We already talked about the glowing fish, the glowfish, that was primarily a novelty item, mesmerizing people with their bioluminescence.
Right, the glowing pets.
Exactly. But it's also created genuinely new aesthetic possibilities in horticulture, developing flowers and colors that simply don't exist naturally. For instance, truly lavender-colored carnations were created using genes from petunias, and the quest for a blue rose, a kind of holy grail for rose breeders for centuries, was finally achieved using genetic engineering, borrowing genes involved in blue pigment production from pansies.
So creating colors nature didn't invent.
Precisely. And then there are even more esoteric examples. Some researchers have actually engineered bacteria, like E. coli, to respond to light in such a way that when they're grown on a plate and light is projected onto them, they produce pigments only where the light hits, effectively creating living black and white photographs, a form of microbial art.
Bacteria making photographs. Okay, that's definitely unexpected.
It really shows the incredible range, doesn't it? From critical life-saving medical treatments and cleaning up oil spills, all the way to making blue roses and bacterial photos. The key insight here is just how versatile this toolkit is. It's not some narrow niche field. It's applicable across this astonishing spectrum of human endeavor, from our fundamental survival needs right through to aesthetic expression and pure scientific curiosity.
It is truly mind-blowing when you lay it all out like that. The sheer scope is immense. But
Yeah.
And it's a big but. As we gain this almost godlike ability to manipulate life, to rewrite its code, it inevitably brings us back to the really tough questions. What about the ethics, the public conversation? How do we navigate this incredibly powerful capability responsibly?
This is absolutely the crucial flip side of the coin. And it's something, as we touched on earlier, that the scientific community itself has been grappling with right from the very start.
Back to the SLMR conference in 75.
Exactly. Scientists recognized the potential risks early on and tried to establish ethical guidelines for recombinant DNA technology. That proactive self-governance was really vital. And today, genetic engineering isn't happening in a vacuum. It's subject to quite rigorous regulation in most parts of the world.
There are laws and treaties?
Oh, yes. There are international agreements, like the Cartagena Protocol on Biosafety. This specifically governs the movement of living modified organisms, GMOs, across international borders, aiming to ensure their safe handling, transport, and use. It shows there's global recognition that this powerful technology needs careful oversight and robust ethical frameworks.
But despite the regulations and the scientists trying to be responsible, there are still big concerns and controversies out there, right? Things that fuel the public debate.
Absolutely. And many of them are legitimate points for discussion. One major area is environmental concerns, particularly around GM crops.
Like what specifically?
Well, critics often raise the fear of gene flow, the possibility that genes from genetically modified crops could spread, through pollen, for instance, to their wild plant relatives.
Creating what they call superweeds.
That's the term often used, yes. The worry is you could inadvertently create wild plants that are resistant to herbicides or have some other trait that gives them an unfair advantage, potentially disrupting natural ecosystems. There's also concern about potential impacts on non-target organisms, like could pollen from insect-resistant corn harm beneficial insects like monarch butterflies? These have been studied extensively.
What about GM animals escaping, like those faster-growing fish?
That's another significant concern, yes. If genetically modified fish, perhaps engineered for faster growth or disease resistance, were to escape from fish farms into natural waters, what impact would they have on wild populations? Could they outcompete native fish? Could they interbreed and spread the modified genes? These are complex ecological questions with potentially serious consequences.
It's worth noting, though, isn't it, that many of these ecological concerns, impacts on biodiversity, pesticide use, gene flow, to some extent, they aren't unique only to genetic engineering. Conventional agriculture has big ecological footprints, too.
That's a very important point, yes. Many of the debates around GM crops touch on broader issues inherent in large-scale, intensive agriculture, regardless of whether the crops are genetically modified or conventionally bred. It highlights the complexities of managing agricultural ecosystems as a whole. But GE does introduce novel combinations and traits which warrant specific scrutiny.
Okay, environmental concerns are one area. What about the really sensitive stuff, editing humans?
Right. Human gene therapy. This is probably the most ethically charged area. As we discussed, somatic gene therapy targeting non-reproductive cells to treat disease in an individual without passing changes to offspring is progressing and generally viewed more favorably, ethically speaking.
Because the changes die with the patient.
Exactly. But germline gene therapy, making changes to sperm, eggs, or embryos that would be inherited by future generations, that is profoundly controversial.
Because you're altering the human gene pool forever.
Precisely. The idea of making heritable changes to human DNA raises deep philosophical and ethical questions. Remember when researchers in China first used CRISPR to edit human embryos back in 2015? Even though those embryos weren't intended for implantation, it caused a huge reaction. Major world science academies called for a global moratorium on any clinical use of germline editing, meaning don't make designer babies yet.
Why such a strong reaction? Beyond the safety concerns.
Beyond the significant safety concerns about off-target effects in embryos, there's the fear of this slippery slope.
The slippery slope from curing diseases to
human enhancement. The worry is, if we start allowing germline editing to cure serious genetic diseases, where do we draw the line? Could it eventually lead to using the technology not just for health, but for enhancing traits perceived as desirable height, intelligence, athletic ability, creating designer babies?
And is that line easy to draw between therapy and enhancement?
Many argue it's actually incredibly blurry. Is correcting genetic deafness therapy or enhancement? What about predispositions to certain conditions? It gets complicated very quickly. And then there are concerns about equity. who would have access to these enhancements? Could it create new forms of social inequality, a genetic divide between the enhanced and the unenhanced?
These are huge societal questions.
Absolutely. And the case of He Jiankui in China in 2018 really brought all these fears to a head.
He was the scientist who claimed he had actually created gene-edited babies.
Yes, he announced he'd use CRISPR to edit the CCR5 gene in human embryos, supposedly to make them resistant to HIV infection, and that twin girls had been born from these edited embryos.
Their reaction.
Universal condemnation from the global scientific and ethical communities. His work was seen as reckless, unethical, medically unnecessary, and dangerously premature, given the unresolved safety issues and the lack of societal consensus on germline editing. It dramatically underscored why extreme caution is needed.
Is germline editing banned then?
Functionally, yes, in most places. Actual germline modification that leads to a birth is currently banned by law or regulation in around 40 countries. And even where it's not explicitly banned, strict guidelines and ethical reviews effectively prevent it for now. Scientists doing basic research on edited embryos almost universally ensure they're never allowed to develop beyond a very early stage, respecting that critical ethical boundary.
Okay, so huge ethical debates there. What about the general public? How well do people feel they understand all this complex science?
That's another really interesting piece of the puzzle. Our sources included a survey looking at public perceptions of genetic engineering, and the findings really highlight a communication challenge.
What did it find?
Well, interestingly, while a good chunk of people, about 54%, so they had some idea about genetic engineering, and another 31% claimed they knew what it is. Over half, 56%, also felt that science wasn't being communicated to them in a way they could easily understand.
So more than half feel left in the dark, basically.
That seems to be the feeling, yes. There's a gap between the science happening and public comprehension.
Does that affect where people get their information from?
It seems to. That communication gap might explain why a significant number of people, almost 27% in that survey, reported learning about genetic engineering primarily from popular culture. Science fiction movies, TV shows, news media, basically the same percentage as those who learned about it through formal education like school or university, which was 25%.
So sci-fi is teaching people about GE as much as science classes.
Potentially, yes.
Yeah.
And if the scientific community isn't effectively translating these complex ideas into accessible language, then popular culture inevitably steps in to fill that vacuum. Which isn't always bad, but it can sometimes lead to misunderstandings, hype, or unnecessary fear based on fictional portrayals rather than scientific reality.
What about trust and control? Do people want oversight?
Very much so. The survey found a clear majority, nearly 69%, believe that genetic engineering absolutely should be controlled by governmental influence. People clearly feel that this powerful, rapidly advancing science needs robust regulation to ensure it's developed responsibly and used in ways that benefit society and align with public values.
And the overall ethical view, is the public generally for or against GE?
It's nuanced. The survey mentioned an online debate that showed opinions were split almost 50-50 on whether GE is ethical overall. However, the specific survey we looked at actually found that most of its respondents viewed genetic engineering as both ethical and useful for humanity.
So maybe a shift in perception, or just different groups surveyed.
Could be both. It does suggest an evolving public perspective, perhaps moving away from some of the earlier, more widespread skepticism. Back in 1976, the Nobel laureate George Wald famously worried whether every scientific advance automatically make our lives more meaningful. There was a real concern that maybe we were meddling too much.
But maybe the benefits are becoming clearer now.
That seems plausible. The tangible benefits, especially the life-saving breakthroughs in medicine like new drugs and potential gene therapies, and the crucial advancements in agriculture for food security, they become increasingly apparent over the decades. This might be steadily shifting some of that earlier skepticism towards a more cautiously accepting or even positive view, provided there's strong oversight. The profound insight here is really that the science itself is only half the equation. Public understanding, trust, and ongoing ethical dialogue are just as vital for responsibly integrating these powerful technologies into our lives and future.
It's been quite a journey. We've gone from those almost magical glowing fish and the scientific sensation of Dolly the cloned sheep, all the way to disease-resistant crops that are feeding millions, and now the truly breathtaking potential to cure devastating human genetic disorders.
It really shows how far we've come. Genetic engineering isn't just science fiction anymore. It's not confined to the lab bench. It's a dynamic, rapidly evolving field that is actively shaping our present reality, what we eat, the medicines we take, and profoundly influencing our future possibilities. It's moved from theory to tangible reality.
So, as we stand here now, gaining this unprecedented ability to literally rewrite the code of life itself, wielding tools like CRISPR that offer such incredible precision and power, what does it all really mean for us, for humanity's future?
That's the ultimate question, isn't it? Are we simply becoming better editors of nature, correcting mistakes, alleviating suffering, improving quality of life through scientific ingenuity, much like a meticulous editor cleaning up typos in a text, using our knowledge for good?
Or is it something more fundamental?
Or are we perhaps standing on the cusp of something even more profound? Are we moving beyond being just editors towards becoming the actual designers of life? Fundamentally redefining what it means to be human and maybe even what life itself can be by taking control of its very blueprint.
It's a huge thought. Are we correcting nature or starting to direct it in entirely new ways?
It's a question with no easy answers. A question for all of us, really, as individuals and as a society, to seriously consider. Something to truly mull over as this incredible science continues its astounding deep dive into the very essence of existence. It challenges our definitions of life, our place within it, and offers both immense, almost unimaginable promise alongside profound, generation-spanning responsibility.